Hi friends, welcome back to NitVed Bioscience. Today our topic of discussion is collagens. We will see the structure, first introduction, then the structure, what kind of the secondary structure present in the collagen, then how the biosynthesis and the post-translational modification will take place, and what are the diseases associated if there is any deficiency or if there is any defect in the formation of the collagens, that we will discuss in the today's class. So first we will see the collagens. From where this name comes? This name comes from the Greek word colla. Colla means glue producing. Glue means which is holding something together. Yeah. So this is the main component of the connective tissue. Okay. It is mostly this collagen protein. This protein is most abundant in the mammals. We can say that around the 25 to 33% out of total body protein. So 25 to 33% of the total body protein is the collagen. Why? Because it is a structural protein. It provides the structure, support, strength, okay, throughout the body. You name any tissue and the collagen is present there. It provides the structural support to the extracellular space of the connective tissue. Okay, it provides the perfect matrix for the skin, tendons, bone, ligaments, connective tissue. Okay, this collagen also present in the nerves, blood vessels, cornea of eyes. Okay, so almost in the all the tissue or all the organs or all the body parts of the body, collagen is present, like teeth, nails. Okay, so collagen is widely present in its main function is it is a structural protein. Now there are the various types of the collagen proteins, like almost 19, or in some books you will read around the 25 to 28, but out of all those collagen types, type 1 to 4 that we will find very oftenly, but out of that type 1 collagen, so out of all the collagen, type 1 collagen is almost 90% of the collagen present is the type 1 type of the collagen. Okay, so this collagen protein, it is unique as compared to the other protein. Why? Because here the secondary structure has a triple helix. Triple helix is there in the secondary structure of the collagen. What is triple helix? Triple helix means the 3 polypeptide chain, they are coiled to each other. Okay, 3 polypeptide chain, these are coiled to each other, that is known as a triple helix. This triple helix is present in the collagen, means unique secondary structure is present in the collagen. 3 polypeptide chains. So just we had discussed that type 1 is most common type of the collagen which is present in the body, almost the 90% of the collagens. So let us take the example of the type 1 type of the collagens. In the type 1 type of the collagen, 2 polypeptide chains, they are similar in the chemical compositions. So this is known as a alpha 1, means 2 alpha 1 chain. Okay, alpha 1, this is also alpha 1, and this is alpha 2. This green color is alpha 2. Why? Because this chain is somewhat different as compared to the other 2 chains. So in the type 1 type of the collagen, which is 90% presents, 2 alpha 1 chains are there, which is similar in the chemical compositions, and the 1 alpha 2 chains are there. They are coiled together around each other and form the triple helix. Okay, so these individual alpha helix chains, these are left-handed. Individual alpha helix, these are left-handed, but after they coil together, Okay, after the biosynthesis, finally we will get the collagens. So this triple helix, these are right-handed, means the 3 left-handed polypeptide chain, they overlap one another, and we will get the super helix or this triple helix, and this triple helix is right-handed. So individual alpha chain is left-handed and this triple helix is right-handed. Okay, so what we had discussed, we will quickly revise. So collagen, we had discussed that collagen, it is a structural protein, it is a fibrous in nature, and it has a rod-like structure. Okay, fibrous rod-like structure, mainly present in the connective tissue, bone, tendon, skin, ligaments, and all the connective tissue. 25 to 33% of the total body protein is a collagen. Okay, and the collagen has a unique secondary structure like triple helix, where the 2 alpha chains are the similar, one is the different, so that is alpha 2 chains. Individual alpha chains are the left-handed, and the triple helix we will get is, it is a right-handed types. Now we will see the amino acid composition, means type of the amino acid presents. So type of the amino acid present are mostly glycine X and Y. Okay, glycine X, Y, where the X and Y can be any other amino acid, but most often it is a proline and hydroxyproline, and the less often it is a lysine and hydroxylysine. Okay, but all the times, every time, every third amino acid is a glycine, means the glycine will compulsorily present in the collagen triple helix. So what is the significance? Why the glycine is a compulsorily present in the collagen structures? So as we can see, it is a triple helical form. Okay, so glycine, when we had studied the structure or of all the amino acid, we had seen that the glycine, in the side chain of the glycine, only the hydrogen is present. Therefore, it is the smallest in nature as compared to all the other all the 19 amino acids. So glycine is the smallest. Because of the smallest, it can occupy the center of this helix. Okay, and this triple helix can have the compact structures, that's why it can withstand the stress or the stretching, it inhibit the stretching because of the forming the compact structures, and that compact structure is formed because of the glycine, because the glycine is the smallest in nature. Okay, small size, only because in the side chain only the hydrogen is present. Therefore, the glycine is there, and the other can be the proline, most often proline, hydroxyproline, and the less often will be the lysine and hydroxylysine. Okay, and any other amino acid can also present here. Now we will see, and okay, so this individual chain, it can have the around the 1000, approximately around the 1000 amino acids are there in the one polypeptide chain, and each turn has a 3 amino acid. Okay, each turn has a 3 amino acid. Around the 1000 amino acid are present in the individual polypeptide chain. Okay, so now we will see how the structure of this collagen triple helix is stabilized. So collagen triple helix is a secondary structure. So like any other secondary structures like the alpha helix, beta pleated sheet we had discussed, where those secondary structure is stabilized by the hydrogen bond. So similarly, here also the structure main bond which is responsible for the stabilized structure of the collagen triple helix is the hydrogen bond. And along with that, so van der Waals interactions, ionic interactions, van der Waals interactions, or the S-sulfur bond, these are also present, sulfide bridges. So this will help in the stabilizing the structures. But how we will see how this will form and what are the various modification it will undergo after the synthesis. So that will come under the heading of the biosynthesis of collagen. So now we will see the biosynthesis of collagens. There are around the 22 genes. 22 genes are there which is responsible for the collagen synthesis. Hence we have the different types of the collagens. So first, like any other process, transcriptions, the first mRNA, mRNA will be formed by the transcription in the nucleus. Then this mRNA will come out of the nucleus and it will attach to the ribosome of rough endoplasmic reticulum. Okay, then first the mRNA will form by the transcriptions. So mRNA will come out of the nucleus, it will attach to the ribosome of rough endoplasmic reticulums. In the ribosome, pre-pro alpha chain or the pre-pro polypeptide, we can write it as pre-pro alpha chain will form. What is a pre-pro alpha chain? Because in the pre-pro alpha chain, some signal peptides are there. Suppose this is a alpha chain. So some signal peptides are there. Okay, so this form on the ribosome, it is known as a pre-pro alpha chain. This signal peptide, it will help to or it will give the direction of this pre-pro alpha chain to come into the lumen of endoplasmic reticulum. Okay, this signal peptide, it will give the signal to this pre-pro alpha chain which is formed on the ribosome to it will give the direction to come into the lumen of rough endoplasmic in the lumen of, yes, it is lumen of the rough endoplasmic reticulum where the further modification will takes place. This is also known as a signal peptide. Okay, once this pre-pro alpha chain, it will enter into the lumen of the rough endoplasmic reticulums, this signal peptide will be removed by the enzyme signal peptidase, and the remaining we will get, it is a pro alpha chain. Okay, this is a pro alpha chain we will get in the lumen of the rough endoplasmic reticulums. This pro alpha chain, before forming the triple helix, it will undergo the various chemical modification that we will see. So these are, first is the hydroxylation, then glycosylation, okay, so first these two. So first is the hydroxylation and the glycosylations. Hydroxylation means on the proline or the lysine, amino acid residue, it will undergo the hydroxylation, means OH group will attach to proline and lysine in the presence of enzyme prolyl hydroxylase or lysyl hydroxylase. Okay, and this process takes place in the for this process to take place, means hydroxylations, vitamin C is required. Vitamin C is very important for this step to occur, means hydroxylation. Okay, therefore due to the deficiency of the vitamin C, there can be the one disease that is known as a scurvy. Okay, so vitamin C is very important. So first is the hydroxylation, proline glycine hydroxylation in the presence of enzyme prolyl hydroxylase or lysyl hydroxylase, and which requires vitamin C as an important cofactor. Then some of the hydroxylated lysine, okay, some of the not all, some of the hydroxylated glycines, they are the glucose or galactose, glucose and galactose, or only the glucose will attach to it. What is the significance of that? It will help in the bond formation, means during the collagen fibril formations, during the forming the complete structures. So during the covalent modification or bond formations, this modification will help. So first is the hydroxylation, second is the glycosylation. If there is a defect in the glycosylations, there the disease occur will be the osteogenesis imperfecta. Okay, So hydroxylation in the presence of vitamin C, if there is any if there is a deficiency of vitamin C, okay, then scurvy. Okay, next step is a glycosylation. If there is a defect or if there is any issue during this step, glycosylation, then the disease will be the osteogenesis imperfecta. Hence we can understand that how much collagen is important to our body. There are various other diseases that I will put in the last of this video. Okay, then okay, after these steps, hydroxylation and the glycosylations, 3 polypeptide chain, it will spontaneously form the coil like structure. Okay, so at the center this triple helix is there, and at the both side some loose ends are also present, both at the N terminus and the C terminus. These are loose ends. Okay, sorry, these are not the helicals at the N terminal. This is just like a loose ends. Something like that, where the sulfide bridges are present. Okay, these are held together by the sulfide bridges. So at the middle, this is having the helical regions, and the N terminus and the C terminus, N terminus and the C terminus peptides are there. So what is the significance of this N terminus and the C terminus peptides? It will help in the pre-maturation of the collagen. Okay, after the formation, this is known as a pro-collagen. Okay, so after the mRNA, okay, so on the rough endoplasmic reticulums, what we get? The pre-pro alpha helix. On ribosome, pre-pro alpha helix. Then after the removal of the signal peptide, which will help in the entry of the pre-pro peptide into the lumen of endoplasmic reticulum, the signal peptide will be removed. We will get the pro alpha helix. This pro alpha helix, after undergoing the various modification like the hydroxylation and the glycosylations, spontaneously they will form a bond and form a triple helix, triple helical structures, where at the center the triple helical presents, and at the N terminus and the C terminal loose ends are present. This is known as a pro-collagen. How this structure is stabilized, just we had discussed, it is stabilized by the hydrogen bond. Hydrogen bond, which is stabilized the structure of the collagen, it is formed between the carboxyl group of the proline of one polypeptide and amino group of the glycine of another polypeptide. Okay, so hydrogen bond is present, and also the disulfide bridges are there, and it will form the pro-collagen. So from the pre-pro alpha helix, pro alpha helix, then the pro-collagen. Then this all procedure will take place in the lumen of rough endoplasmic reticulum, in the endoplasmic reticulum. After that, it will transfer to Golgi complex. This pro-collagen in the Golgi bodies or Golgi complex. Where it will assemble into vesicles okay. Suppose this is a triple helix with the loose ends. Okay, now it is easy to distinguish. So this pro-collagen will packed into the vesicles of Golgi bodies. So all this modification, now this takes place intracellularly. After the Golgi bodies assemble into the Golgi bodies, this pro-collagen will come out of the cell. Okay, and then some extracellular modification will takes place. So now after coming out from the cell, this pro-collagen it will be acted upon by two enzymes, N-procollagen peptidase and the C-procollagen peptidase. What is the function of those enzymes? It will cut this N-terminus loose end of the procollagen and the C-terminus loose end. Only the triple helix will remain here, and this is known as a tropocollagen. Okay, so from procollagen, now we will get the tropocollagen. Procollagen means triple helix and the loose end at the N terminus and the C terminus. And what is that? Is procollagen. Sorry if I am making any mistake. Procollagen means triple helix with both N terminus and the C terminus loose end. When it will be removed by the enzyme N-procollagen peptidase and the C-procollagen peptidase, it will remove this loose end, and what we will get is known as a tropocollagen. Means procollagen will convert into the tropocollagen. As soon as this tropocollagen we will get, so this will arrange in a ordered staggered way. Suppose this is one tropocollagen, so it will have the another here. Okay, if we indicate it with just a simple line, so one tropocollagen, another, so it will arrange in a staggered way, which will arrange around means the distance will be around the 3/4 of the adjacent molecules. So it will arrange in a staggered way, spontaneously it will arrange in a staggered way, and bond formation will take place between the tropocollagen. Now means linkage will take place here. Okay, and we will get here the collagen fibril. Collagen fibril. Okay, enzyme involved here is the lysyl oxidase, and the action of these enzymes will take place in the presence of copper. Now we will get the collagen fibrils, and many such collagen fibril, it will come together and we will get the collagen. Okay, so now let us quickly revise what we had studied so far. The collagen is synthesized by the specialized cells. These are known as the fibroblast cells. The first is the transcription will take place, then the mRNA will come out of the nucleus. This mRNA will then attach to the ribosome of the rough endoplasmic reticulums, and there will be the synthesis of the pre-pro alpha chains where the signal peptide will be present. This signal peptide will direct the pre-pro alpha chain into the lumen of the endoplasmic reticulums. In the lumen of endoplasmic reticulum, after the removal of the signal peptide, we will get the pro alpha chain. This pro alpha chain then undergo the modification like hydroxylation and the glycosylations. After glycosylation, the triple helix formation will take place, which is known as a procollagen. Procollagen has a triple helix at the center and the loose end at the N terminus and the C terminal, which then transfer to Golgi bodies. Then after the coming to the Golgi bodies, then through the process of exocytosis, it will come out of the cells where the cleavage of the N terminus C terminal will take place. We will get the tropocollagen. Then there will be the cross linking in the presence of the lysyl oxidase. We will get the collagen fibril, and many such collagen fibril, they will come closer and we will get the finally the collagen fiber. And these are the some of the example of the diseases associated with the defects in the synthesis of collagen. Thank you.